today we will discuss about Escherichia coli. Now, first of all, what is this organism? This is a gram-negative bacilli and it is commonly found in the environment, in the soil and in water. Furthermore, this organism also is able to survive within the human body and also along with other animals. And predominant region where it survives is the mucous membrane, and perhaps the biggest source is the gut. So how this organism has come or is started being associated with living or animals? It was originally present in the environment, especially in the topsoil, subsoil and in water. So most of the animals were grazing animals. So when they used to eat the grasses, this organism entered into the intestines. And throughout the period of evolution, this organism has also evolved along with the evolution of human beings. And today, as of today, this organism is both a pathogenic organism and a commensal. Okay, that means it can cause various diseases. Also, it is a part of the commensal flora of the gut. Now, here comes the interesting thing. Why we have to study of this organism? Like Staphylococci, the organism we had studied previously, this organism has potential to cause infections almost anywhere in the body. Okay. Furthermore, the genetic predisposition of this bacterium allows it to modify and become resistant to the commonly used antibiotics to such an extent that it can also transmit the genes responsible for producing proteins which can degrade antibiotics from one to other. Okay. So hence, in today's scenario, this is a very difficult organism, especially in the case of drug resistance that we are seeing today. Now, this organism along with other organisms have been classified into a group of bacteria called as Enterobacteriaceae. As the name suggests, this group of bacteria are commonly associated with the entrails of human beings. That means these organisms are commonly associated with the intestines. Now, there are few common characteristics of all these organisms. Obviously, there are some exceptions. Nevertheless, let us look at some common characteristics. First is they are all gram-negative bacilli. Second, they are all aerobes and facultative anaerobes. Now, what do you mean by facultative anaerobe? Look at this picture. In last class, I also told you this is a liquid culture media and it contains various organisms. Now, the top part is open, that is exposed to air. The air contains oxygen. If an organism is an obligate aerobe, that means it cannot survive without oxygen. What will happen? The growth will be seen predominantly in the top half because the oxygen concentration decreases from top to bottom. Similarly, at the other end of the spectrum, we have aerotolerant anaerobes. Anaerobes won't be growing in this media because some oxygen is dissolved in it. If anaerobes are exposed to oxygen, it will die. So some of the bacteria can still survive. Those are called as aerotolerant. That means they can survive the atmospheric concentration of oxygen. They will not replicate, multiply, but they can survive. Now coming back to the obligate anaerobes, they will grow at the bottom of the test tube. And what the facultatives will do? They can grow almost throughout. They will prefer oxygen, but even if there is lack or decreased concentration of oxygen, they can grow. Okay. This is the reason why Enterobacteriaceae can survive in the intestine. Intestines is not an anaerobic environment. However, the concentration of oxygen is very low as compared to environment outside the human body. Okay. Next, these organisms are non-fastidious. Non-fastidious means they can survive on minimal nutritive requirement. Okay. They can utilize the simple nutritive elements like sugars and other elements. Hence, in the laboratory, when we try to culture them, these organisms can easily grow on basal media. What is the basal media? It is an example of basal media. That means we do not enrich them with other things. For example, blood agar is an enriched media. It is enriched with blood. Okay. So these all Enterobacteriaceae can easily grow on basal media. Next part is they can ferment glucose to produce acid with or without gas. Now glucose is a very basic form of sugar. So all the Enterobacteriaceae they can ferment glucose. It may be associated with production of gas. Some will produce gas, some will not produce gas. So when then we test in the laboratory, like you can see in this picture, the first one they have indicators. If there is growth, what will happen? The glucose will be utilized with production of acid. This acid will change the pH and there will be change in color. As you can see over here, there is no fermentation seen with glucose. On the next test tube, there is fermentation, but there is no production of gas. And on the last test tube, you can see there is a small pocket of gas that is collected in a very small tube within the test tube. This is also called as the Durham's tube. Okay. Then we can interpret in the second test tube, we have fermentation of glucose without gas, and on the third test tube, we can say fermentation of glucose with production of gas. Understood? So all bacteria which are part of Enterobacteriaceae can ferment glucose. Now when I am saying ferment glucose, what do you understand by fermentation? What is fermentation? Right. If the sugar is utilized with presence of oxygen, we call it as oxidative utilization. If it is utilized without the use of oxygen, we call it as fermentative utilization or fermentation. Okay. Remember, these are facultative anaerobes, hence they can ferment. Another important part is they can reduce nitrate to nitrite. This is one of the features by which it can survive in the intestine and also in the soil. And this test is utilized as one of the characteristics of Enterobacteriaceae. Next is they all are catalase positive. Please recall what I have told you about catalase test in the last class. Catalase enzyme degrades hydrogen peroxide to release nascent oxygen and water. This enzyme is present in all aerobic and facultative anaerobic organisms. Because they are facultative anaerobe, they have catalase enzyme. Obviously, there are exceptions. Finally, they do not produce oxidase enzyme. Hence, on testing oxidase, it will be negative. And the last common feature of Enterobacteriaceae are most of them are all motile with peritrichlous flagella. Now, what is the peritrichlous flagella? It could be unitrichous, lophotrichous, amphitrichous, and there are examples of peritrichous. Most of the bacteria belonging to Enterobacteriaceae are motile by peritrichlous flagella. There are some exceptions also. Whenever there is a rule, there are exceptions to the rule. So let us recapitulate what are the important points of Enterobacteriaceae. These are important. First is they are all gram-negative bacteria. They are and they are aerobic or facultative anaerobe. They can grow on basal media. They all ferment glucose with or without production of gas. Then they reduce nitrate to nitrite. They are all able to produce catalase enzyme, oxidase negative, and finally they are all motile by peritrichlus flagella. These are the generalized rules. There are exceptions. Now, since there are many bacteria, the whole Enterobacteriaceae have been classified into 8 tribes. Among these 8 tribes, as you can see, named after the important organisms like Escherichia, Edwardsiae, Salmonellosis, Citrobacteriaceae, Klebsiella, Proteaceae, and Urinary. Among them, we will be discussing about Escherichia. In the laboratory, when we are trying to identify an organism, how do we identify any organism? How can we identify any organism? Hmm? Ferment. Anything else? This is a general discussion. I'm not teaching anything. How can I identify any bacteria? जोड़े बोलो. Stain. Okay. Anything else? See, the points which we use to identify any bacteria first will be its morphology. When we say morphology, that means what is the shape, the size, the kind of stain it can take up. The stains they do not take up. If it is not able to take up any stain, we call it as negative staining. If it does take up any stain, we call it as positive staining. Okay. So these all are morphological features. Next, anything else? Another part is metabolomics. That means study of metabolism of a bacteria. Because the bacteria will grow, propagate, that means it can utilize some sugars, chemicals, etc., etc. That means those are metabolized. So when it is metabolized, we can test each and every type of material. These are called study of the metabolomics. Anything else? We can study the genes that are present in the bacteria. Obviously, the bacteria will have DNA. We can study the DNA. One type of bacteria will have a different sets of DNA as compared to the other bacteria. This is called as a genomic study. Fourth one would be what comes out of a gene or DNA? Protein. Now, the proteins are the building blocks of life. One particular bacterium is different from another because it has different sets of protein. Why it has different sets of protein? Because it's got a different sets of DNA. So by the help of the proteomics, we can also differentiate. So these are the 4 main fundamental points by which we can identify. Okay. So all the stainings, morphology, these all come under morphological study. Then we come to the metabolic study. So one of the very easy way to differentiate these Enterobacteriaceae is how they grow on a particular agar called as MacConkey agar. Why this is important? Remember, I already told you all Enterobacteriaceae utilize glucose. So if I use a media containing glucose, I cannot differentiate between various types. That is the reason why in MacConkey there is lactose. So this differentiates the Enterobacteriaceae into 2 broad groups. One is called as lactose fermenters and one is called as non-lactose fermenters. See, the media contains very small amount of glucose which will allow the bacteria to survive, but it will have a larger amount of lactose. So some particular groups of bacteria will utilize the lactose to form acids. That acid will cause change in color and we can identify them as lactose fermenters. Some group of bacteria will not be able to utilize the lactose and there will be no formation of acid. So there will be no change in color of the corpus. We call them as non-lactose fermenters. These are the 2 broad group of classification. Now look at this picture. On a MacConkey plate, on the left hand side, we have an organism. What can you see? These are pink color. And what color colony is this? No, it has got the same color as the media. Look at the background of the media. It is all the same color. That means the colonies are colorless. Whereas on the left hand side, every colony is pink in color. Now, why they are pink in color? Because we have put an indicator in the media. This indicator will change the color whenever there is presence of acid, that is change of pH. Why does the pH changes? Because this particular bacteria can utilize lactose, whereas this bacteria cannot. Understood? Hence, we call it as this one as lactose fermenter and that one as non-lactose fermenter. Understood? Okay. Now, let us look at Escherichia coli itself. It will have all the features that we have already discussed about Enterobacteriaceae. Most importantly, let us look at how it was named. It was discovered by Escherichia in 1885, hence named after him. And why coli? Because it was found in colon. Next, these are found in the gut of human and animals, and because they are found in human and animals, what will be the natural cycle? It will come out to the fecal matter, and from fecal matter to soil. In the soil it will survive. Then again grazing animals will eat the grass. From there again into the intestine. This is the normal cycle, it survives. Now let us know the virulence factors of E. coli. Now we have understood that this organism causes human disease. It is both a commensal, but yet it can cause human disease. That means it will have some factor which will help with causing the disease. What are these factors? These are called virulence factor. First factor is called, it has got 4 antigens. One is called somatic antigen, one is called flagellar antigen, capsular antigen and fimbrial antigen. Somatic means body. Now look at this picture. The somatic antigen, that means body. The lipopolysaccharide is present on the cell wall. So that antigen is also called as O antigen. Okay. Again, E. coli will have a capsule around it. The antigen that it produces because of the capsule is called as K antigen. Because in those days in the German language, capsule was written with K, hence it is called as K antigen. Okay. It will also have fimbriae, those are called as the F antigen. Another set of special types of fimbriae are present on particular subset of E. coli which are called as pili or pilin protein. What do? They helps the bacteria to adhere to mucous membrane. This is the reason why only some subset of E. coli will be able to cause urinary tract infection through the ascending route. When I say ascending route, that means from urethra to bladder, because a normal human being will micturate. So the bacteria has to move against the flow of the urine. The only way it can move in, because if it has something to latch onto the mucous membrane. Those are the pili proteins. Not all subtype of E. coli will have pili protein. Those subtype which has the pili protein will be able to cause urinary tract infection through the ascending route. Others would not be able to do so. Okay. And finally, the bacteria itself can produce in some proteins which we call them as toxins. Those are not toxins to the bacteria, those are toxins to the human body. Hence, they are called toxins. So, these toxins can be either enterotoxin. Enterotoxin means the toxins which have local effects on the intestine. That's why they are called as enterotoxins. Other would be cytotoxic necrotizing factor. That means these toxins can affect almost all type of cells locally. Cytotoxic because damage to the cells. These toxins have been further divided into predominantly 3 types. Initially, it was seen this toxin had two parts. One part was heat-labile, another part was heat-stable. That means if you take the bacterial suspension and heat it slightly, one part of the toxin was destroyed, but other part was not destroyed. Hence, it was called as heat-labile toxin, and this one is called as the heat-stable toxin. Another type of toxin was also seen, which was similar to a toxin produced by bacteria called Shigella. It was called as Shigella-like toxin or verocytotoxin. Why these are important? Look over here. The heat-labile toxin is also called as enteroform produced by enterotoxigenic E. coli. I will discuss it later. It resembles cholera toxin. Just remember this now. When you will study about cholera, you will understand more about it. What will it cause? It will cause watery diarrhea. If you have got any idea, cholera produces watery diarrhea as a toxin mediated diarrhea. The heat stable toxin, this also causes watery diarrhea, but its mechanism of action is different. And finally, the Shiga-like toxin or the verocytotoxin, this inhibits the 60S ribosomal subunit. That means it inhibits protein synthesis. Because it inhibits protein synthesis, it acts on the local cell, causing cytotoxicity. Remember, all human cells are constantly undergoing protein synthesis. If protein synthesis is stopped, the cells will undergo lysis. One more important point that you should remember, these two toxins, the heat-labile and the heat-stable, they are transmitted by plasmid, whereas this is bacteriophage coated. Means it is transmitted by viruses which affect bacteria and hence can transmit rapidly. Okay. Now, what are the clinical manifestations? Most commonly we see is in the community settings, you see urinary tract infections, especially in females. It causes UTI. However, I have already told you not all strains of E. coli can cause UTI. Only particular strains which have particular pathogenicity will cause. Those are called as uropathogenic E. coli. So, other than that, what other disease it can cause? It will reside in the gut. Local action because of the toxins, it will produce diarrhea. Now, because it produces various types of toxins, we have many types of outcome. These are called as enteropathogenic E. coli, enterotoxigenic E. coli, enteroinvasive E. coli, enterohemorrhagic, enteroaggregative, and diffusely adherent E. coli. Okay, we'll discuss later on. Other than these two, it can cause almost any type of infections like abdominal infections, respiratory tract infections, especially in the hospital settings. Then meningitis in neonates in the peripartum period, wound and tissue infections, even osteomyelitis and sepsis in the hospital setting because of the intravascular device related infections. Now, if we need to diagnose a particular infection, how will we need to diagnose? First, we need to collect the sample. That will depend on the type of infection that we see. If we are suspecting this is a case of urinary tract infection, the sample would be urine. If we are suspecting it is a case of bacterial sepsis, the sample would be blood. After we take up the sample, the first step would be to make a microscopic examination, where what do we will find? We will find gram-negative bacilli. And on culture, we will find lactose fermenting colonies on MacConkey agar. Next step is, I have already told you most of these bacteria are motile. So, we need to test the motility. Motility testing can be done. E. coli are motile. Coming to metabolomic studies, what will be the findings? It will be catalase positive, oxidase will be negative. Next tests are indole, citrate, urease, TSI. These are the metabolomic study that is commonly done in the manual method. The same metabolomic study is done by automated method like Vitek, or we can do the proteomic study with the help of the multitoph. And the last part of the diagnostic part is testing for antimicrobial susceptibility, because at end of all testing we need to treat the patient. After identify the organism, we need to know which antibiotics it is susceptible to. Now, important biochemicals which you should know about this bacteria we will discuss. First is the indole test. What is the principle of this test? The bacteria will produce tryptophanase, which will act on tryptophan to produce indole derivatives. You don't need to remember the indole derivative. What you need to remember, some Enterobacteriaceae can reduce tryptophan to indole derivatives. If that is formed, then the test will be positive, as evident by formation of a indole ring. I think you can see the red color ring on the left hand side. This is called as indole test. E. coli will be positive for indole. That means it can produce tryptophanase. Next is citrate test. The principle of citrate test is that in this media there is no carbohydrate source, no amino acid as energy source. The only source is citrate. That means some bacteria which can survive even in harsh environmental conditions where nutrition is very scarce can survive. These bacteria can utilize citrate as a sole source of carbon. Whatever we eat, what is the main energy giving source? It's carbon. So, some bacteria can utilize citrate as a sole source of carbon. For example, Klebsiella, I think you have heard about this organism. It is also part of Enterobacteriaceae, but it can utilize citrate as a sole energy source. But E. coli cannot. So, in this case, we will have citrate not utilized. Okay. In examinations, please do not say citrate positive. Either citrate will be utilized or it will not be utilized. So, if we look at this media, this is the citrate media where the organism has grown, but there is no color change because citrate has not been utilized. On the left hand side, there is change in the color to bluish color because utilization of the citrate. Utilization of citrate will end up giving pyruvic acid. This acid will cause change in the pH, resulting in change in the color. Next important test that you should know is urease test. Now, why urease test is important? Because in the environmental conditions, the animals will urinate in the water bodies and on land. The urea that is present is harmful to the bacteria. So, some organisms which can survive in the environment will be able to degrade the urea with the help of an enzyme urease. So, we test in this whether a bacteria can produce urease or not. Okay. Klebsiella can produce urease, but E. coli cannot. So, in this case scenario, urease test will be negative for E. coli. That means urease not produced. If urea is produced, what will happen? It will degrade urea to ammonia and water, forming ammonium hydroxide, which is basic in color, will increase the pH. That means there will be change in the pH. There will be change in the color. As you can see, the right hand side test tube, there is change in color from the left hand side. This is the principle of urease test. Have you understood? So, if any bacteria is able to produce urease, it can survive in the environment, like Klebsiella can survive. Now, coming to the last biochemical test, as far as you people are concerned, it is called as the TSI test, or triple sugar iron media. So, when we say triple sugar, that means there will be 3 sugars. The three common sugars are glucose, lactose and sucrose. Now why glucose is given? Because purpose of lactose and sucrose is to differentiate various types of organisms. But initially we need to give some carbohydrate source so the bacteria can survive. That is the glucose. Normally we will have 1:10:10 percentage of glucose, lactose and sucrose. What will happen? Look over here. First let us look at the lactose fermenter. First it will utilize the glucose, and when the glucose is exhausted, which it will get exhausted very quickly because we have 1/10 in amount, it will start utilizing the lactose. When the lactose will be utilized, what will happen? It will form acid. When the acid formation occurs, the media will change its color into yellow. Okay, this we represent as A by A. Now why A by A? Because if you observe carefully, the test tube has two parts. One is the butt and then is the slant. The slant, why two part? Because the slant has lot more air being exposed. So the reaction that we say is A by A is slant by butt. Now suppose if the organism is again able to utilize, after the lactose get exhausted, it can utilize the sucrose. What will happen? It will again produce acid. Thereafter also it will form the reaction as A by A. But if it fails to utilize sucrose, what will happen? It will start utilizing the amino acids present in the media. When it will start utilizing the amino acids, degradation of amino acid will produce ammonium hydroxide, which is basic in nature, and it will cause change in color in the slant. It will go back to the original reddish color because the acid has been neutralized. That reaction we call it as K by A. And in case of non-fermented, there will be no acidic change. Look at this picture. There is no change in color. Okay, we can say this is a non-fermenter. When we say non-fermenter, it utilizes only glucose. Over here what we can see, it is K by A, that is basic by acidic reaction. This will happen when the organism will utilize lactose, and thereafter in the slant it is utilizing the amino acids. If it again utilizes the sucrose, it will keep on producing the acid. Then we can see acidic by acidic reaction here also. We call it as A by A reaction. If the organism cannot utilize anything, it will be K by K, basic by basic reaction. Some groups of bacteria in Enterobacteriaceae will be able to produce a gas called as H2S or hydrogen sulfide. This hydrogen sulfide would react with iron that is present in the media, triple sugar iron medium. It has got iron, it will react with the iron to form ferric sulfide. That ferric sulfide will get precipitated to form black color. But please remember, this reaction will only occur in acidic environment. So this reaction, even though if it is black in color, we will say it as K by A with production of H2S. Have you understood? Yes or no? মনে তো হচ্ছে না কি স্যার ঘুম পেয়ে গেল বেসিক ফান্ডামেন্টালটা বোঝা গেছে আমরা এখানে জাস্ট দেখছি একটা হচ্ছে কার্বোহাইড্রেট মেটাবলিজম আর অ্যামিনো অ্যাসিড ডিগ্রেডেশন ইন প্রেজেন্স অফ অক্সিজেন ঠিক আছে one single test tube test can determine whether it's a lactose fermenter or not, sucrose fermenter or not. ঠিক আছে now that we know E. coli is a lactose fermenter, we can say its reaction will be A by A. that is acidic by acidic. Okay. Now let us recapitulate what the biochemical test at least required for you people is that it will be catalase positive, oxidase negative, then indole will be positive because it will be able to produce tryptophanase. The tryptophan will degrade to form indole derivatives. Then citrate cannot be utilized by E. coli, but other group of Enterobacteriaceae can utilize. It cannot produce urease. Finally, in triple sugar ion medium, we will have A by A with or without production of gas, but it does not produce H2S. Okay. Now coming to the treatment, most of the treatment will be based on the antimicrobial susceptibility reaction test. However, in today's time, we need to be aware of multi-drug resistant organism. That is a particular strain which is resistant to many groups of at least 3 or more classes of antimicrobial drugs. Since you have not been taught antimicrobial agents till now in your pharmacology classes, I think so. So we'll discuss all these resistance and susceptibility part later. Now coming to diarrheogenic E. coli. The first is enteropathogenic. You don't need to mug it up. Try to understand, then it will all be very simple for you. What is the name of this particular E. coli? It is called as enteropathogenic. What does it cause? It causes infantile diarrhea mainly in form of outbreak. Now enteropathogenic means it causes direct pathogens. How? It will adhere to the intestinal mucosa and leads to disruption of the brush border of the epithelium. And when the brush border epithelium is disrupted, what will happen? Absorption will not be occur and it will cause diarrhea. Now why it causes infantile diarrhea? Any idea? Why is it more commonly seen with infants? Even in immune competent, not all infants are immune compromised. No, it is mainly due to during the infancy. Initially, the child is exclusively breastfed, then our normal food is started to the child after 6 months. So when outside food is given, if this bacteria gains access earlier, it will cause the diarrhea. Slowly and steadily, when the child will start eating normal food, there will be change in the gut flora. When the gut flora, many types of bacteria start colonizing the intestine, then this bacteria will not be able to cause the disease more frequently. Hence, after the 1 year period, this disease is less commonly seen. Next is, that was enteropathogenic, cause it causes direct pathogenesis to the brush border of the epithelium. Now comes enterotoxigenic E. coli. Toxigenic T, remember traveler's diarrhea. Traveler's diarrhea is also called as Delhi belly. There is also a movie called Delhi belly. I think somebody might be knowing about. No? Okay. Traveler's diarrhea was very frequent in Delhi, hence it is called as Delhi belly. Why? Because it forms toxins. What kind of toxin? This is the heat-labile and heat-stable toxin. I have already told you 2 types of toxins. These are preformed toxins. So whenever you are going out, eating so-called outside food in not so good hotels and roadside stalls, after eating, next day you end up having diarrhea. Most commonly, it is caused by enterotoxigenic E. coli. Okay. Next type is called entero-invasive. Now try to understand the word invasive. What do you understand by invasive? Invasive মানে কি হতে পারে? Internal মানে from the gut it can invade into bloodstream. This E. coli is genetically and pathogenically similar to Shigella. It produces the toxin and it will lead to invasion, means disruption of the mucosal membrane, ulceration of the bowel, and if the ulceration deepens, what will happen? The capillaries will be eroded. When the capillary is eroded, what will happen? The blood will come out along with the fecal matter. That is called as dysentery. And because it is causing disruption and ulceration of the bowel, there will be inflammatory reaction evidenced by huge number of pustules in the fecal matter. And if there is ulceration in the intestine, what will happen? Intestine irritate হলে কি হবে? হ্যাঁ, আরে ফুড অ্যাবজর্পশন তো পড়ে, তোমার একদিন ফুড অ্যাবজর্পশন না হলে তোমার কিছু এসে যায় না। What will happen if there is inflammatory reaction going on the intestinal wall? হ্যাঁ, এটা তো শুধু এপিথিলিয়ামে নয়, আরো ভিতরে ঢুকছে, তাহলে ইরিটেশন হলে কি হবে? আরে আলসার তো হয়ে গেছে, হবেটা কি? Exactly, the migratory motor complex gutter, that will be triggered and there will be cramping abdominal pain. ঠিক আছে? So the typical description of dysentery কি হবে? Patient will come and say there is severe cramping pain in the abdomen, and after that the patient will go to the laboratory. There will be very small fecal matter with blood, and when we look under the microscope, RBCs and huge number of pustules. This is called as invasive. Okay, এতদূর বোঝা গেছে? প্যাথোজেনিক হলো, টক্সিজেনিক হলো, ইনভেসিভ হলো। এবার হচ্ছে এন্টেরোহেমোরেজি। This is by a special type of E. coli called as O157H7. This causes outbreaks mainly by contaminated food, and it produces Shiga-like toxin mediated, and we can identify by sorbital MacConkey agar, তোমাদের লাগবে না। মেইন জিনিস হচ্ছে it will produce dysentery, and aftermath of this disease will lead to causing hemorrhagic colitis or hemolytic uremic syndrome. আমি হেমোলাইটিক ইউরিমিক সিস্টেম আজ এখানে ডিসকাস করছি না, আই থিঙ্ক প্যাথোলজি তোমাদের পড়ানো হবে। ঠিক আছে, ইটস আ microangiopathy that may lead to hemolytic uremic syndrome. Only thing that you should remember, enterohemorrhagic E. coli will cause dysentery, and this is caused by Shiga-like toxin or verocytotoxin. This will cause outbreaks. Remember যখন টক্সিনটা বলেছিলাম কি, it is transmitted by bacteriophage, hence local outbreaks very common. Another type is called as enteroaggregative E. coli. It is produces the EAST toxin producing enteroaggregative. These are and diffusely adherent. These two তোমাদের জন্য বেশি ইম্পর্টেন্ট না, as far as you people are concerned. মেইনলি তাহলে আমাদের কি হচ্ছে, প্রথম হচ্ছে enteropathogenic. Enteropathogenic will affect the local epithelium, disruption of the epithelium causing diarrhea. Next will come as enterotoxigenic. This is caused by preformed toxin. heat-labile হতে পারে, heat stable হতে পারে. It will cause profuse diarrhea. ঠিক? Next is enteroinvasive. Invasive means will cause ulceration and bleeding, leading to dysentery. Last is enterohemorrhagic. It also causes dysentery, but by producing Shiga-like বা verocytotoxin. These are the four main types. ঠিক আছে, এবার তোমরা বলো যদি diarrhea or dysentery is caused by E. coli, what kind of treatment would you recommend? Obviously antibiotics will kill the bacteria. কোনটায় অ্যান্টিবায়োটিক দেবে, কোনটা দেবে না? না না, আমার কোশ্চেন খুব সিম্পল। তোমার দেখো ইপি মানে এন্টেরোপ্যাথোজেনিক, এন্টেরোটক্সিজেনিক, এন্টেরো ইনভেসিভ আর এন্টেরো হেমোরেজিক। অ্যান্টিবায়োটিক কোনটায় দেবে, কোনটায় দেবে না? হেমোরেজিকে দেবে আর ইনভেসিভে দেবে, আর দেবে না? শিওর? This is the most important thing that I want to stress upon. এন্টেরোপ্যাথোজেনিক মেনলি কিভাবে হয়? ছোট বাচ্চাদের হয়, যেখানে অন্য অর্গানিজম গাটে পলিফাইড করার আগে এই পার্টিকুলার ইকোলাই গ্রো করেছে, যার জন্য ডিসরাপশন হচ্ছে। আউটকাম কি হবে? ম্যাল অ্যাবজর্পশন। At one point of time other bacteria will grow and most of the time the child will grow out the illness. Okay, আমরা যেটা ম্যাক্সিমাম করতে পারি, ম্যাল নিউট্রিশন হলে বাচ্চাকে বারে বারে ফিড দিতে হবে, বাট অ্যান্টিবায়োটিকের বিরাট কিছু রোল নেই, বিকজ ওটা হচ্ছে লোকাল ডিসরাপশন। যতক্ষণ ব্যাকটেরিয়াটা এপিথেলিয়ামের উপর থাকবে ডিসরাপ্ট করবে, তারপর ব্যাকটেরিয়াটা মরে গেলে বাচ্চা ঠিক হয়ে যাবে। There is no need for antibiotics. Next come enterotoxigeny. It is by preformed toxin. You can give many antibiotics, but the disease will run its course. Why? Because the toxin is already being absorbed in the intestine. There is no role of antibiotic. Enterotoxigeny কি করে? Travelers diarrhea করে। Any role of antibiotics in travelers diarrhea? হবে কি কোনো রোল আছে? নেই তো। Next time when you have travelers diarrhea, don't take antibiotics. Important antibiotics should be given in case of invasive and hemorrhagic. In both these types, you will have patient manifesting with dysentery. যদি পেন, টেনেসমাস, ক্র্যাম্পিং, পেন এগুলো থাকে, দ্যাট মিনস ইনভেসিভের দিকে যাচ্ছে। ওগুলো কমনলি অ্যাসোসিয়েটেড না থাকলে হেমোরেজিকের দিচ্ছে। হোয়াট এভার ইট মে বি কেস, there is a high probability that the bacteria can gain access into the bloodstream because of the damaged capillary endothelium. Hence, you have to give antibiotic in this case. Okay? Ideally টেস্ট করে দেওয়া উচিত, অবভিয়াসলি হ্যাভ টু বলে আমি ওইভাবে বলছি না, টেস্ট করা উচিত। সো এইখানে টেস্ট যদি আমাকে ইনভেসিভ আর হেমোরেজিকে টেস্ট করতে হয়, কি টেস্ট করতে হবে? স্টুল টেস্ট করতে হবে। স্টুল টেস্টিং তোমাদের পরে পড়ানো হবে, এখন বললাম। আচ্ছা, তাহলে কি হলো? ট্রাভেলার্স ডায়রিয়াতে হচ্ছে কি মেনলি হচ্ছে তোমার এটা সেলফ লিমিটিং। আচ্ছা, এন্টারো অ্যাগ্রিগেটিভ যদি থাকে তাহলে হচ্ছে ইমিউন কম্প্রোমাইজ যদি থাকে তখন দিতে হবে। ওকে, সো দিস ইজ ব্রিফলি অ্যাবাউট, দাও শেষ হয়নি, about antimicrobial therapy in case of intestinal effects. Extra intestinal effect যদি হয়, what we'll have to do, we have to treat according to the antimicrobial susceptibility report. Okay, it may be UTI, it may be sepsis, it may be meningitis, whatever it is, based on the antibiotic susceptibility. One important part is, many a times especially young females will have UTI and it is not possible to test sometimes. So two commonly used antibiotics are prescribed without testing. One is a nitrofurantoin and the other, as far as people concerned, nitrofurantoin. Okay, that can be given without testing. Okay, if it is of community origin. Otherwise we need to test in all cases, especially because of the increase of resistance that is seen with antibiotics. One more important practical part that I want to discuss is, all throughout this discussion we have said that most of the bacteria of Enterobacteriaceae are motile. That means we need to test motility. Now I am just going to discuss motility testing very briefly. How can we test whether a bacteria is motile or not? Solid media. Anything else? Hanging dog preparation. Anything else? Participate koro, participate na korle hobe. This is a group discussion. তোমরা ডিসকাশন না করলে তো লেকচার হয়ে যাবে তাহলে। মোটিলিটি আমরা কিভাবে দেখতে পাই? আচ্ছা, মোটিলিটি, why does the bacteria becomes motile? Flagella. That means there is an appendage which helps the bacteria to move. তাই তো? তাহলে first part is we can observe under the microscopic examination. What we can observe? Microscopic appendage, in this case the flagella, right? কিভাবে দেখতে পাবো? Flagella stain. আমরা যদি ব্যাকটেরিয়া ফ্ল্যাজেলাকে ডিরেক্টলি স্টেন করি, then we observe them under microscope, we can by flagella staining. Anything else? আর কোন রকম ভাবে আমরা মাইক্রোস্কোপিতে দেখতে পারি? হলো ফ্ল্যাজেলা স্টেনিং এবং ফ্লুরোসেন্ট নন ফ্লুরোসেন্ট যেভাবে হচ্ছে ফ্ল্যাজেলা স্টেনিং, গল্প শেষ। আচ্ছা, তুমি যে হাঁটতে পারো, দ্যাট মিনস ইউ আর মোটাইল। আমি যদি তোমাকে বলি প্রুভ দ্যাট ইউ আর মোটাইল, তুমি কিভাবে প্রুভ করবে? দ্যাট মিনস ইউ উইল শো ইউর মুভমেন্ট, তাই তো? তাহলে ডেমনস্ট্রেশন অফ মোটিলিটি, মোটিলিটিটাকে আমি এখানে আমরা মোটিলিটি ডেমনস্ট্রেট করছি না, উই আর অনলি অবজার্ভিং দ্য ফ্ল্যাজেলা, দ্যাট ইজ অ্যাপেন্ডেজ অফ মোটিলিটি। Another microscopic method is demonstration of motility. This demonstration of motility can be done directly under the microscope. This method is called as hanging rock preparation, by which we can demonstrate motility under the microscope. Okay, আর কিভাবে আমরা ডেমনস্ট্রেট করতে পারি? আর কিভাবে ডেমনস্ট্রেট করতে পারি? একটা হচ্ছে, দেখো, ফর আ মোমেন্ট তুমি উঠে দাঁড়াও। হি ইজ মোটাইল, হি হ্যাজ অ্যাপেন্ডেজেস, 2 legs by which he can move. Either I can demonstrate that he has legs, or I can show a video of you that he is walking around, or I can show that he has moved from this place to that place. That means he has already moved, that is displacement. So we can study the displacement. This displacement study can be seen with the help of culture media. Okay, suppose if I take a test tube, and on this test tube there is a media. Okay, there is a very small U-shaped tube over here, and this tube has a media. Now I take small inoculum of the bacteria and inoculate this point. এইখানে মিডিয়াটা নিচে আছে, উপরে নেই। এইখানে আমি মিডিয়া ইনোকুলেট করে দিলাম। The next day, if I am able to demonstrate that the same organism is over here, how has it moved over here? It has moved through this tube. That is, the bacteria has displaced from one place to another. This is called as the Craigie tube method. Okay. Similarly, on a solid agar plate, which we can see very commonly with a highly motile bacteria like Proteus. Suppose I have inoculated the bacteria over here. After overnight culture, we can see there is swarming growth. Swarming means the organism has moved from the original inoculated area to the other areas. That is, the bacteria has displaced from inoculated to non-inoculated area. These are the methods by which we can demonstrate motility. Okay. What will be the best method? Best and the easiest method is a direct demonstration. That is hanging drop preparation. You don't need to do a stain, you don't need to inoculate, you don't need to wait for one full day to demonstrate that the bacteria has moved. The best method for easy diagnosis is hanging drop preparation. How do we do hanging drop preparation? We take the organism on a test tube with a liquid culture media, inoculate it, and then incubate for about 1 to 2 hours, so that the bacteria can multiply in huge number. And if you do remember the bacterial growth phase, initially there will be a lag phase, then log phase, then the plateau phase. মনে পড়ছে? আমি যদি এই ফেজে দেখি কি হবে, ব্যাকটেরিয়া খুব কম থাকবে, দেখতে অসুবিধা হবে, অনেক সময় নাও পেতে পারি। এই ফেজে যদি দেখি তখন কি হবে, হাফ ব্যাকটেরিয়া ডেড, half bacteria alive. Dead bacteria is non-motile. Dead bacteria doesn't move. তাহলে এখানে আমাদেরকে লাভ নেই। তাহলে আমাকে দেখতে হবে এই লগ ফেজের এখানে, যেখানে ম্যাক্সিমাম ব্যাকটেরিয়া আর অ্যালাইভ অ্যান্ড গ্রোয়িং। গ্রোয়িং হলে তাহলে কি হবে, দে উইল বি মোটাইল। দিস ইজ দ্য রিজন ওয়াই অ্যাবাউট 1 to 2 আওয়ার্সের মধ্যে আমাকে ইনকিউবেশন থেকে করতে হবে। ঠিক আছে। নেক্সট we will take a glass slide and a cover slip. On the fore end of the cover slip wax drop দিতে পারি, অথবা আমাদের একটা এই সার্কুলার বিড থাকে যেটা কমনলি ওই প্লেডো বা ক্লে যেটা বলে, সেটা দিয়ে একটা সার্কুলার ইয়ে করতে হবে। In the center we will place 1 drop of this liquid media after 1 to 2 years. Then we'll invert it on this glass slide. তাহলে এইরকম থাকবে। এইবার যে এজটা আসছে, we have to observe this edge. প্রথমে 10x এ এজটা ফোকাস করব, তারপরে 40x এ অবজার্ভ করব। 40x এ যখন আমরা দেখব তখন আমাদের ভিউ পয়েন্টটা কি হবে, সাপোজ দিস ইজ দ্য ভিউ পয়েন্ট, we will be observing this edge. This water drop যেহেতু আমরা উল্টো করে রেখেছি, it is actually এই যদি কভার স্লিপ হয় এটা এইরকম ঝুলছে, হ্যাঙ্গিং ড্রপ। বিকজ ইট ইজ হ্যাঙ্গিং, হোয়াট উইল, ওয়াটার ড্রপের ওয়েটটা তো নিচের দিকে, তাহলে ম্যাক্সিমাম ফোর্স কোথায়, দিস পয়েন্ট। কে ধরে রেখেছে, ওয়াটারের সারফেস টেনশন। তাহলে সারফেস টেনশন সবচেয়ে বেশি এখানে, সবচেয়ে কম সারফেস টেনশন কোথায় হবে, যেখানে কভার স্লিপে টাচ করছে। তাহলে ব্যাকটেরিয়া যদি মোটাইল হয়, ইট উইল মুভ ফ্রম আ প্লেস অফ হায়ার সারফেস টেনশন টু লোয়ার সারফেস টেনশন। তাহলে সবসময় এই এজের কাছে চলে আসবে। সেই জন্য আমরা এই এজটাকে 40x এ ফোকাস করবো। এই 40x এর এজটা যখন ফোকাস করবো তখন দেখবো ব্যাকটেরিয়াগুলো এইখানে মুভ করছে। এবার ডাইরেক্টলি যদি আমরা ডেমনস্ট্রেট করতে পারি দ্য ব্যাকটেরিয়া ইজ মুভিং on that particular age, we can say the bacteria is motile. This is hanging drop preparation. ঢুকেছে কিছু মাথায়? এবার যদি পরীক্ষায় জিজ্ঞেস করে মোটিলিটি টেস্টিং বলতে পারবে তো? হ্যাঙ্গিং ড্রপ is one of the method of motility testing. Other is we can demonstrate the appendage commonly by flagellar stain, demonstration of motility itself, অথবা movement of the organism by Craig's tube method or swarming. There are various other methods. Okay. Well, that is all about E. coli today. In your practical today, you will be given a culture plate of E. coli on MacConkey agar. First, you need to do a gram stain, look at the biochemicals, and hanging drop দেওয়া হবে। সেই hanging dropটা তোমরা দেখবে মোটিলিটি।